Yes.
Did you share your slide?
Sir, it's now live,
Uh,
sir. You can go, sir.
yeah. Yes, sir.
No, yeah, share your slides, sir.
Yeah, I have already shared, sir.
Yeah, it will come. It will come. It's downloading. Yeah, it
Yeah.
came.
Yes, sir.
Just 1 minute, because exactly by 2 o'clock we will start. Now time is
Yeah.
1:59.
Yes, yes, yes.
So it's 2 o'clock. Shall we start? Dr. Logesh Deb, can you hear me?
Yes, sir. Yeah, yeah. Can you hear me, sir?
Yeah, it's 2 o'clock. Shall
Yeah.
yeah, I can hear you.
Yeah.
Shall we start?
Yes, sir.
Right. Okay. Thank you. Good afternoon. Myself, Professor D. Karthikeyan, Principal and Convener, SNS College of Pharmacy and Health Sciences, Coimbatore, Tamil Nadu. Yesterday, we had a webinar which was presented by Dr. Pawan Kumar, Director, R&D, Dr. Reddy's Lab, Hyderabad. And tomorrow, also we'll have the third session will be presented by Dr. P.K. Lakshmi, former Director of Drug Information Center, Karnataka State Pharmacy Council, Bangalore, and Professor and Head Department of Pharmaceutics and Pharmacy Practice, G. Pularity College of Pharmacy, Hyderabad. Today's webinar will have the schedule like welcome the speaker and participants to this 3-day national webinar, and introduction of speaker will be done by Dr. D. Karthikeyan, and then the presentation followed by
ಇದಕ್ಕೆ ಲಿಂಕ್ ಪಡಿಸಿ ಏ ಇದಕ್ಕೆ ಲಿಂಕ್ ಪಡಿಸಿ ಆಯ್ತು
send the chat messages. Speaker will respond. Finally, vote of thanks by Dr. Kumar Nalasivan, professor and coordinator. On behalf of SNS College of Pharmacy and Health Sciences, it's my proud privilege to welcome the today's speaker, Dr. Logesh Dev, Scientist D, Institute of Bioresources and Sustainable Development, Ministry of Science and Technology, Government of India. And I also welcome all the participants for this today's webinar. Let me introduce Dr. Logesh Dev. He was born in Mirza village of Tripura, India on 13th February 1978. Completed his school education in Kairit Bikram Institute, Udaipur, Tripura. Completed diploma in pharmacy in 1999 under a Board of Examination Authority, Drug Control Department, Government of Karnataka, Bangalore. Completed B.Pharm in 2003 and M.Pharm Pharmacology in 2005 under Rajiv Gandhi University of Health Sciences, Bangalore, Karnataka. He was awarded PhD degree in the Faculty of Pharmaceutical Sciences by Jawaharlal Nehru Technological University, Hyderabad. Apart from his academic courses, Dr. Deb completed short-term hands-on professional training courses on animal cell cultures in CDRI Lucknow, HPLC, HPTLC in Natural Remedies Private Limited, Bangalore, Flow Cytometric Techniques, C-Camp, NCBS, Bangalore, and refresher courses on basic biostatistics, experimental animal handling, and as well as ethical aspects and the collection, cultivation, management of medicinal and aromatic plants. And before he entered in this government service, he worked in teaching industry, In 2008, he joined Institute of Bioresources and Sustainable Development, Impal, Manipur, which is a National Institute of Department of Biotechnology, Government of India, as a scientist B, and continued his research activities on ethnopharmacological research and discovery from the traditional medicine of Northeast India. Presently, he is working as scientist D in IBSD, Sikkim Center, Gangtok, and engaged in ethnopharmacological survey, collection, and identification of bioresources used in traditional folklore medicines of Northeast India, and pharmacological and toxicological evaluation of traditional formulations, individual components, active components to justify the traditional claim and to develop economic, safe, novel therapeutic agents. His research also accompanies value addition to those bioactive components for commercial exploitation. He also worked as a visiting scientist in Cytokine Research Laboratory, Department of Experimental Therapeutics, the University of Texas, USA, under DPT Biotechnology Overseas Associateship Program. He is a life member of 11 different national and international professional academic societies. He has published more than 70 research articles and review articles in national and international journal with a high impact factor. He also published about 41 abstract in national and international conferences, seminar proceedings, delivered more number of innovative lectures and two book chapters also he has presented. He has an Indian patent in his credit. So today we have such a wonderful, knowledgeable speaker. Once again, I welcome Dr. Logesh Deb. Now I hand over the session to Dr. Logesh Deb. Dr. Logesh Deb, please proceed. Thank you.
Thank you, sir. Thank you very much for the introduction. I'm Lokesh Deb. I'm working in Institute of Bioresource and Sustainable Development, Department of Biotechnology, Government of India, in Sikkim Center right now. I'm basically working on ethnopharmacological research and drug discovery here. So in the same aspect, today I'm going to tell you my experience, what I came across during my work here. So in that context, I have designed this title, Ethnopharmacological Research and Drug Discovery, its prospective and challenges. So let's know first some familiar terminology. What actually this traditional knowledge? It's age-old practice of community associated with its survival. And there are thousands of human communities on this earth, even in our country, even in other countries of this of the world. And each and every communities has their own skill and age-old practice in several walks of life, like handicraft, food, medicine, textile, dance, sports, agriculture, custom means costumes, etc. In fact, it is cultural practice of those communities. And ethnopharmacology is a scientific study of ethnic groups and their use of drugs in the sense therapeutic agents. And ethnomedicobotany is a tools which breeds a relationship between the plants and mans to prevent and cure the human ailments. And as you know, all of you, that Northeast India, it comes under two mega biodiversity hotspots, that Indo-Burma as well as the Himalayan biodiversity hotspots in the world map. Therefore, huge variety of plants growing in this region, and these are all valuable plants, and indigenous peoples of these regions, they are totally depend on these naturally growing vegetation, either for food or for their medicine. And indigenous medicinal plants growing in this northeast region of India is useful folk medicine used by the peoples of this region. It means here I'm talking about one of my research components. What I have done in Manipur. It's another state of Northeast India. As you know that there is total 8 states in Northeast India: Sikkim, Arunachal Pradesh, Assam, Meghalaya, Tripura, Mizoram, Manipur, and Nagaland. So earlier I was in IBSD Imphal, means Manipur Center, which is our head office. And in Manipur, now actually 13 districts are there. Those days, 9 districts was there. All 9 districts I have covered, means done the ethnopharmacological survey and documented all traditional knowledge of traditional practitioners. And so far, you can see on this screen that number of traditional practitioners or folklore healers, means in different districts they share their traditional knowledge with us. And there are number of challenge in this ethnopharmacological research, because you cannot convince each and every folklore healers for sharing their knowledge. So even you cannot force them. If they are willing to share, then only you will get that materials. So far 89 traditional practitioners in Manipur, they have shared their traditional knowledge with us. And you can see in first phase, some clips I have presented here. So they have a traditional way of treatment, traditional way of diagnosis, traditional way of physiotherapy, each and everything. Even in photograph, you can see that in right top, these two patients of paralysis and this gentleman, Mr. Tohal Meiti, Mr. C. Tohal Meiti, these are his inpatient. So they are from far off places, and he is doing physiotherapy with the help of some plant products, and people are getting cured. Even paralysis patients, those are not getting sense even in organs like limbs, hands or legs. And after doing such physiotherapy with the traditional way, as well as with the natural products, they are getting cured. And that's why this gentleman, he is getting inpatient even from far away places. And in below, you can see that patient, he is actually cancer patient, Mr. Sele Asor. He is a traditional practitioner. In extreme right, you can see that gentleman with that Diplopoda species of insect. He is making a medicine out of this species, and means treating the cancer. Even this gentleman is a multiple myeloma cancer patient, and he was discharged from hospital and back to the home by saying that hardly 6 months he will survive. But this gentleman saved this patient at least for more than 4 to 5 years. So this is miracle of our traditional healthcare practice. Even in that, you can see that gentleman means in middle, he is doing bone setting, and he has in traditional way of hospital, and means he is successfully creating the bone fracture. So with that natural products. And in extreme left of the middle, you can see one lady. She is means actually diagnosing patient like our classical means now modern doctors. So I want to pass a message that traditional healthcare practice is not a superstition, not an unscientific thing. There are science yet to explore, and means they have all kind of therapeutic components like physiotherapy, diagnosis, treatment, etc. And there are some more clips in third phase of survey. So finally, Means nowadays we are talking about the COVID-19, and means it's a heroic pandemics in all over the world, and even in our country the case are increasing like anything day by day. And if you see these three plants, Hartania cordata, Colocasia esculenta, Urtica dioica, even means peoples of Northeast India, they are taking as a vegetable since long. Even practitioners are making traditional medicine out of those uh, plants. And if you see that graph in context of our country, so compared to the mainland India, in northeastern India, that COVID cases are comparatively less, and death rate is very negligible, maybe due to such practice. And this Hotenia cordata, Colocasia esculenta, Urtica dioica, these plants are tested against the SARS-CoV-1 in on the 2008, 9, even 2013 in China. So means we have asset with us, and our traditional healthcare practice can provide such remedy. With this slide, I want to apprise you that how we are losing such valuable traditional healthcare practice. If you see that age group of the peoples here, means youngsters are not much interested. Even those are old traditional practitioners, their son and daughters, they are studying outside of the states, and they are doing some other work. They are not at all interested in their traditional practice. That's why the preservation of such valuable knowledge is very much important. What we are doing means in documentation. And one more significance of this ethnopharmacological survey is you can see those gentlemen, they are not no more with us, they are no more on this earth, they all died. So means initially I have documented their traditional knowledge. Now it is preserved, very much preserved with us. Even their family members don't know what they used to practice and what are the medicine they used to make and how. All those documentation is only available with us. We have videography, we have written documents, we have audiography, means audio visual, as well as the written documentation we have. And of course that this program we have done in collaboration with Traditional Practitioners Association of Manipur. So that is so called Aponba Manipur Maiba Maibi Furuf. So of course the one copy of the documentation also available with them, so it will be even available for next 100 years. And apart from such traditional knowledge, we also try to understand that what is the genesis of that traditional healthcare practice. And we came across with some mythological story, means in Manipur, that once upon a time, a sun god actually means prescribed some plants to treat the person Laithong Puthiba. And according to that story, actually, mistakenly, the sun got injured that person by kicking his horse, what he was riding that time. And that injured Puthiba was treated by the prescribed medicines of sun god. And in that called is Siddha Hidat Toret, and people are believe that that may be the genesis of that healthcare practices in Manipur. And you'll astonished to see that means prescribed medicines for 7 days in a week, means prescribe 7 medicines. So and if you see that Ningtokha, that means Monday in Manipuri language, Meithil language, Ningtokha means Monday, and he prescribed the Tinospora cardifolia, that is called in Manipuri, it's called Ningtokholli. There is resemblance in the name of the day as well as the name of the plants, and in that way for 7 days 7 medicines was prescribed according to that mythological story. And if you see at in the uh, eyes of science, so those all 7 medicinal plants, they have all means analgesic activity, antibacterial activity, anti-inflammatory activity. Means if you will think they one trauma patient or one accidental injured patient, if you want to treat such patient, we have to give painkiller, we have to give anti-inflammatory, we have to give antibiotic, then of course anxiolytic. So whatever the drug regime we need for treatment of a injured patient, all kind of drugs are present in that prescription. So that indicates that even traditional healthcare practice also has science. Apart from that, we have came across with some ancient literature that is called Hida Kiyachal. That is actually formulary of the traditional medicines of Manipur, and even copper plate, stone inscription, in which they have described about the treatment of different diseases. And later on this in 1983, that in Meithilong, in Meithilong language, in Bengali script, that Hida Kiyachal was translated again, and means these are the ancient literature actually which indicates that scientific healthcare practices of Manipur. And you can see that valley is called Tosiam in Tamenglong districts of Manipur. So that place in 70 kilometers of surrounding, you won't get even a single modern medicine shop or modern medicine hospital. The peoples of this region is totally depend on the traditional healthcare practices. In different villages, different practitioners are there. They are the doctor for those particular village, and they totally depend on the natural product. So, in summary, I would say that in Manipur, 9 districts to have covered, 89 traditional practitioners, and about 1,223 formulation we have documented, which contain 546 plant, 42 animal parts, as well as the organic and inorganic materials like salt, sugar, molasses, etc. There's 40, and with that 1,223 formulation, they are treating 67 different disease. And this is our first articles, which is published in the Journal of Ethnopharmacology, and second article yet to publish in Indian Journal of Traditional Knowledge. And not only that, documentation, after documentation, all collected data we have analyzed with the statistical tools. So it means our survey data we have compared with the that published literature, because across the world people's also doing such kind of survey, and they are reporting that use of those particular medicinal plants for different disease in different region. So those data we have collected, and according to the different formula of the statistics, we have compared our data with the available literature, published literature data. And then we found, you can see within the circle, our maximum data are within the circle. That means within the statistical significant region, and only 2-3 data is going out. That means whatever survey we have done is unique kind of information we have gathered in that. And what next after survey? If you see that one proposal is submitted to the National Health Policy in 2002. Dr. Devnath is Dr. P.K. Devnath. He was a renowned pharmacologist of our country, but right now he is no more he died in last year. So he first apprised our government that what is the difference between the traditional medicine and synthetic drug discovery. That means why reverse pharmacology of the drug development is useful for speedy discovery of the drugs. So you can see that means in traditional practice, those drugs, those therapeutic agents or natural products are under human trial for long time. And means our work is we have to do the toxicological study, animal study, standardization, and that good drug or active principle we have to do, and within 2-3 years we could come up with a therapeutic agent. But if you talk about the synthetic drugs, it will take minimum 12 to 15 years, because we have to start from the food drug, then active principle, then animal study, toxicology, and then human trial. So in reverse way, you can go in case of the traditional products, the traditional medicine. So therefore, means this model is very much useful for the drug discovery. And after our survey, we used to compile our data in such a way that one commonly used medicinal plants we can select in that model. So you can see in this slides that different traditional practitioners, and if we'll tabulate those different formulation, what documented, and what are the components present in that. If there is three formulation, if all three formulation contain the total 7 or 8 components, so in that way we can write. And then means first plants, whether it is present in the first, second, or third formulation or not. In that way we can check, and you can see that first component, that is first plant, of course, that number one is the plant which is present in all three. That means it is commonly used by the traditional practitioners for a particular disease. For example, means anti-inflammatory disorder, sorry, anti-inflammatory agent for inflammatory disorders. So in that case, we can select that plant which is commonly used, and we can go for the pharmacological and toxicological screening. And another way of selection is by literature survey. We can survey for those traditional medicines, or means particular plants, in PubMed, Science Direct, Google Scholar, and then we can prioritize those plants, how many work is done, whether means in which aspect those plants were explored, whether pharmacological study has been done, or chemistry has been done, and how many papers were published. Accordingly you can make the priority list. So the star means we have prioritized, means given that priority to that particular plant. So, and means now what is the challenge in that? You have done the survey, you have documented the things, and means traditional practitioners, they share their traditional knowledge with you. And now main challenge is to identify that particular plant. Sometimes that traditional practitioners may not able to show you that plant, because maximum plants are annual plants. So in some season they are not, they are dying. So traditional practitioners may have only dry materials with them. So with that you cannot identify. Even means Calodendron colibroken, if I will take example of that plants, a Calodendron informatum, Calodendron, each and every plant, means in Calodendron genus, each and every plant is looks like same. And even in traditional means regional dialects, regional language, it's called Uthap. Uthap means all kind of Calodendron plants. But Calodendron colibrochinium is very much effective in the hypertension, but Calodendron informatum is not effective in the hypertension. So in that way, if you see these slides, so all are pedi, all are oryza sativa, but they are different. So to understand that difference, we have to take the help of the taxonomist. So taxonomy is very much importance in ethnopharmacological research to for proper identification of the plant materials, and means even in language that in different regional dialects, some place the Sareka Osoka, that called as Ashoka, in some places these plants longifolia also called as Ashoka, and if you see the appearance of these plants is almost same. Therefore, proper identification is very much challenging in that ethnopharmacological research. And one more thing, phytochemistry. So, if we will talk about the Brahmi, some places, even in South Indian region, means even in means North Indian region, people knows that Bakupa Munieri is the Brahmi. But in Western, means East and Northeast states, the Brahmi means Central Asiatica. And if we will go for the phytochemistry, then we can understand the differences. In case of the Bacopa, Bacoside A and B is the means active constituents, and aceticoside B and aceticoside AC is active constituents for the central acetica. Even the pharmacology also important tools in the ethnopharmacological research, because I am sharing with you a story in Manipur. It's in 2008. One of the traditional practitioners, he cured a patient. Actually, that patient's having a lumps in the neck area. So with this decoction of these plants, he cured that patient. And according to the traditional practitioners, he thought it was a cancer. So he spread that news that he is able to cure cancer with these plants. And overnight, these plants become famous. It came in the print media, electronic media, and people started purchasing that decoction in means from that reason. Ultimately, when we did that pharmacological screening, we have seen that it does not have any effect against the cancer cells. Of course, it is very good antibacterial agents. So even in traditional practice, we came across with some information that that plant during the Second World War, whatever the horse injured during war, so those get the infection, so that infection treated with these plants, so that means it is a antibacterial active antimicrobial agents, not anti-cancer agent. So therefore pharmacology also important tools for the ethnopharmacological research. So with that experience, we have designed our research strategy, that traditional knowledge, which is anecdotal evidences, then means natural product chemistry laboratory, pharmacology, as well as the bioinformatic laboratory have to work together to get some value-added products out of the traditional medicines, and the same traditional medicines after valuation we can back to the society for that therapeutic use. So means with that strategy we have started to work. A few example I will present you here. These three gentlemen, Mr. Birchum, Mr. Dhoju, and Mr. Adin, they are the traditional practitioners in different regions of Manipur. I have collected three different formulation from them. Uh, of course, all three formulations are for treatment of the joint pain. And one by one, each formulation, each components we have screened, and first we go for the in vitro screening for all isolated compounds, all components present in those formulations. And then one bioactive formulation, one bioactive components we have selected, and we have done the in vivo test, and means even in molecular level we have done. And finally, there out of these 15, we could get the 3 bioactive components, and with that 3 bioactive components, we have reformulated one anti-arthritic formulation formula, which contain resinolic acid, gerumban, as well as the citral, and means and it is very much effective as effective those traditional medicines. And also we have done phytopharmacological, we will also establish the phytopharmacological relationship of that formulation. We made that formulation in the different dose level with the different combination of this bioactive components, that of course that crude medicine means crude extracts only we have used, but we standardize that crude extract in such a way that we can means quantify the active constituents which is responsible for that particular means ailments, means treatment of that particular ailments. So accordingly, we found that if we will increase the jurumban, so it's very much effective against the pain, as well as if you see that formulation 9, we increase that citral, and means we that replace that resinolic acid, still we get very good result against pain. So in that way we have formulated in a medicine out of the traditional knowledge. And with this means slides, I want to invite you all that we have a webinar series in our institute every Saturday from 6 to 8, and it's a international platform. You can go through our website www.ibsd.gov.in. So every Saturday 6 to 8 you can join all international speakers as well as the national speakers. You can listen here in context of COVID, and title of this webinar series is Reimagine Ethnopharmacology. Our honorable director, Professor Pulak Kumar Mukherjee, he is actually moderator for this webinar series. I would like to acknowledge our director, Professor Pulak Mukherjee, for his guidance and encouragement. I also thankful to my colleagues in all center. We have head office in Manipur, we have center in Meghalaya, Aizawl, Mizoram, Meghalaya, Shillong, Mizoram, Aizawl, as well as the Sikkim, Gangtok. And I'm thankful to all my colleagues as well as the our supporting staffs. I'm extremely thankful to our research scholars, our students. Without their help, we may not able to do such kind of program out of this busy schedule. And thank you very much to you all for patience hearing. And I would like to thanks organizer for their invitation and giving me a golden opportunity. Thank you very much.
Thank you, sir. Now the session is open for discussion.
Yeah.
Yeah. Any message, chat message, whether you have received?
No.
Nothing is there, no? Okay, sir. Can you hear me now?
Yes, yes.
I think there is no queries.
Yeah.
So on behalf of students, I would like to ask you one doubt. Means
Yes.
what the novelty or the criteria or things required to patent the natural product or natural components? So because nowadays patenting the natural product is very very tough. It's
Yes,
a
sir.
very tough job, it seems. So how to patent it? How to patent? What are the expectation?
Actually, sir, novelty means
Because we are telling novelty. Novelty, when we have novelty, we can go for patent. But what
yes,
type
sir.
of novelty we can give from the for the natural products or the natural components?
No, in case of its efficacy, if it, efficacy which is not yet reported anywhere, that if you have some efficacy which is better than that available agent, then you can go for that. It means you have to prove that your product is unique compared to the available agents, available therapeutic agents. And even it is possible, sir, means pattern you can do for natural product also, there is no problem. But things is that there should be a novelty in the efficacy, in the formulation, or in the even in packing system. So
Okay, thank
stability,
you.
in case of stability.
Hmm. Okay, sir. Thank you, sir. As there is some network problem, Dr. Kumar Nalasavan is not available to present the vote of thanks in live.
Yeah,
I
no
will do
problem,
that
sir.
job.
Yeah.
Doctor, yeah, he's facing some this network problem. He is
Yeah,
in
yeah.
remote area. That's what. And
Mhm.
Dr.
Yes, sir.
Logesh Dev presented a webinar on ethnopharmacological research and drug discovery perspective and challenges in a simple but more informative, knowledgeable, and understandable way. Now the participants may have the idea about the traditional medicines used in the Northeast India and the treatment method, and how the IBSD is working to analyzing the traditional medicines for the benefit of present and future generation. I hope this will be beneficial for all the participants. I thank the speaker for accepting our invitation beside his busy schedule, particularly now the pandemic COVID-19 period, because he is working on an assignment related to COVID-19. Thank you, Dr. Logesh Dev.
Yeah.
I,
Welcome, sir.
I I thank all the participants for their continuous support. And last but not least, I thank my team members for their dedication, hard work, and valuable support to make the national webinar a tremendous success. Tomorrow we'll have the third session between 2 to 3 p.m. This is for kind of information. And thank you, one and all. Thank you. Thank you very much, sir.
Thank you, sir. Thank you very much.
Thank you. Thank you. Thank you.